Welcome, ladies and gentlemen, to another exciting edition of your APES Notes. We're going to talk about two major topics today, biodiversity and the effects that humans have on the populations of other organisms on the planet. So sit back, get out your notes, and get ready to fill in some blanks. So as I briefly mentioned in class, biodiversity is dealing with the number of species that you find. Whether you're talking about the biodiversity of a particular ecosystem, a particular area, or of the entire planet. There are several aspects that contribute to biodiversity, namely genetic diversity, the range of all the genes, the sum total of all the alleles, the different variants of genes that you have in a gene pool, both the ones that you can actually see in the population and the recessive traits as well, the sheer number of species or species diversity that you have in an area, and the ecosystem diversity, which is the diversity of the habitats that you have within a defined geological space. Before we go further, let's just talk numbers for a moment here. Now, the most recent estimates have stated that there are approximately 9 million eukaryotic species on the planet Earth. This includes plants, animals, fungi, and those rarely talked about organisms, protists. There is an unknown number of bacteria on the planet. Some estimates range from anywhere from 10,000 species to several million species. We are constantly finding new species of bacteria. About 50% of these estimated species are found in tropical rainforests, which only covers 7% of the planet's surface. And out of the approximately 9 million estimated species that exist, only about 1.5 million have been classified and have scientific names. So there's still a lot of the Earth's diversity that we have yet to explore and yet to discover. These next two slides are just giving some visuals on the estimated number of eukaryotes that exist on the planet and the percentage of those that we believe that we've discovered. So if you'll look at the top, you'll see a circle or a pie chart that is shaded with the colors of the continents or the shapes of the continents. The gray shaded region is the percentage of eukaryotes that we believe we've cataloged after about 250 years of cataloging organisms. You'll notice it's a small percentage, about 14%, and the remainder of the species are uncatalogued, undiscovered as of yet. That's also visualized on the bottom. The gray shaded squares that you see represent those species that we know, that we have classified, that we have studied, and the red squares represent those that have yet to be classified. Just in case you were wondering what approximate percentage of the various kingdoms of life remain to be discovered, here are a couple of images to show you that. So we have the pie charts of the various classifications. You'll notice Animalia, there are approximately 6 million species left to discover. I'm assuming those are probably mostly invertebrates or smaller organisms. It's kind of easy to count your mammal species. We're going to skip over Chromista. It's not a major classification. Fungi, there are approximately 500,000 species that have yet to be classified. Plantae, there are about 82,000 species. You'll notice it's less than a quarter of the plants that exist. And protozoa, which are protists, which are smaller, mostly single-celled creatures. We've only discovered about 3/4 of those that are thought to exist. Now, many students have asked me throughout the years, why is biodiversity important? Biodiversity provides a lot of things. First of all, there's just the fact that it provides beauty, it provides aesthetics, it provides something wonderful to look at and to experience in nature. Beyond that, if you're looking for more concrete things that biodiversity provides, it provides food. There are people who do gather food from the environment. It provides food for other organisms as well. We get wood, lumber for fire, for building. We get energy from various living things. We get free recycling of nutrients, free recycling of materials. Also, ecosystems and the various organisms that inhabit them help purify air, water, and soil. They provide natural pest control. And with all these undiscovered species out there, or with extended study of species we've already discovered, you can discover new materials that can be used in anything from engineering to clothing, and can discover new medicines as well. There are factors that will both increase biodiversity and decrease it. So things that will increase biodiversity are having a number of diverse habitats, When you have disturbances in habitats, after those disturbances, when things are recovering, the biodiversity level in that disturbed area will increase. If you don't have a lot of variation or changes in the environment, if you have a lot of trophic levels, more complicated ecosystems and interspecies relationships, actually being in the middle stages of succession, and when you have increased evolution of new species. To decrease biodiversity, some of the factors that you'll see causing that are environmental stress, more extreme environments where it's harder for organisms to live, limitations to the resources that organisms live, so that's why you'll see generally biodiversity is lower in deserts, there's not enough water, extreme amounts of disturbance or changes to habitats, importation of species, that's an anthropogenic thing, and then geographic isolation will also cause decreases in biodiversity. And just since I mentioned evolution, evolution is related to biodiversity, and that speciation, or the evolution of new species, will increase the biodiversity of an ecosystem, whereas extinction will decrease the biodiversity of an ecosystem. Now we've talked about the general concept of biodiversity, let's look at some of the effects that humans have on biodiversity. All of the organisms that you see pictured on this slide are either endangered due to human activity, or they've become a nuisance as they've been introduced into new ecosystems and are causing damage to local ecosystems. So the last thing I mentioned on the previous slide is that you can introduce new species to an ecosystem. These are called invasive species, or otherwise known as non-native species. So these are foreign to an ecosystem, which means there are no natural population controls for them. You probably don't have natural predators in the area, and there aren't any of those limits that would keep the population from growing without any sort of limit to it. What will happen is then these species can outcompete and crowd out native species. This is easy for these species to do because they tend to be R strategists, the ones that are invasive species. They can reproduce very quickly and they can experience very rapid population growth. We're going to discuss 3 particular cases of invasive species, all 3 of which are found in the United States. So the first invasive species that we're looking at, the giant Asian carp, is actually several different species, but they've invaded the same general area, and they're all in the carp family. Giant carp were first brought in from China in 1831. They were brought in as fish food. They're sometimes still used as bait. And they've spread since then throughout the entire Mississippi River system. Not just the Mississippi River itself, but all of its tributaries. There are very large worries that if the giant carp species end up entering the Great Lakes and can establish their populations there, that they would cause the local fisheries, which produce a lot of jobs and a lot of money, would then collapse. So there's been a lot of worry in the government about how to keep the giant carp from reaching the Great Lakes system. The amount of carp that's found in some of stretches of the Mississippi are so great that I have a quote up here. It says, in sections of the Illinois River where the carp has taken hold, it makes up 9 out of every 10 pounds of living material, and that includes both plant and animal. So it's 90% of the biomass that you find in those areas. This map shows the distribution of carp in the river systems of the United States as of April 2013. There are actually real-time maps that are located online where people can actually report their carp sightings. More is done to try and detect where carp are located. They'll actually take readings from water sources, they'll take samples, and try to determine if any of these giant carp species, if any of their DNA has been introduced into these lakes or rivers. So this is a pretty major threat. You can see not only has it spread throughout the entire Mississippi River system, but it's also spread to lakes and other areas west of the Mississippi as well. Don't worry, if you want to see giant carp in action, there are many YouTube videos of them, and we'll happen to watch several of them in class. Apologies to y'all, this is one of several slides that I added in after I sent off the notes to be printed, so this is something that you're going to have to write by hand. I recommend that you pause the video at this point, write this stuff down, and then go ahead and listen to the audio. So as I said, a big concern is keeping these giant carp species out of the Great Lakes. To that effect, the U.S. government has actually created several electrical barriers to try to block these fish from entering the Great Lakes. A couple of other methods for reducing the carp population or trying to control this carp population is bait trading. So trading out for people who actually use these giant carp as bait, so that they're using some other fish species that's not as invasive, to more strictly enforce transport and shipping regulations. There's also electrofishing, where an electric current is introduced to the water. It momentarily stuns the fish so you can capture them. And some people are starting to use a pesticide called rotenone and just blanket it over a river or over a lake, but there are worries that when you have high enough concentrations of this rotenone to kill the giant carp, it'll kill a lot of other fish species as well. So none of these are, by themselves or even together, extremely effective methods of making sure the carp species can stay contained. A few things to notice on this map. So this map is showing you areas of the Chicago River where you can have potential entry points for the carp to enter Lake Michigan, and from there to get a foothold into the Great Lakes. I'd also like to point out on the bottom left-hand corner, the area that's labeled electric shock hazard area. That's where the electric barriers have been set up by the U.S. Army Corps of Engineers. And we're going to take a little closer look at what those barriers look like on the next slide. This is a section of the Chicago River where several electrical barriers have been set up to try to repel carp to make sure they don't migrate into the Great Lakes. The direction of river flow is going to be from the right side of the screen to the left side of the screen, and it's going to be away from the Great Lakes into the Mississippi River. However, the carp can swim upstream. So if you'll notice, the bullet point labeled number 1 shows a bunch of carp trying to swim upstream. As they encounter the electrical fields at points 2 and 3, they are repelled by those. Though there are some steel bands that are going through the bottom of the canal, and you have an electric charge going through them. And as you go further along this, further upstream, what you're going to find is an increase in the strength of the electrical field, so that hopefully the fish will be repelled. You need to have 2 of these fields to try and make sure that none of the fish are getting through, and they're actually rebuilding a new one. So there's a lot of maintenance work to be done to keep this up to date, and it doesn't have 100% efficiency either. Speaking of aquatic invaders, the next species we're going to look at, the zebra mussel, is also a large problem in the same part of the country, in the Mississippi River area. Zebra mussels, as you can kind of see from this picture, are small clam-looking species. Mussels are related to clams. So what you see that looks like gravel on this picture is actually a large amount of zebra mussel shells that are just on the beach of a body of water. What you can see from this map right here, each red dot pinpoints an area where zebra mussels have been found. The green dots, if you're wondering about that, are the sightings of another invasive species of mussel, and the yellow spots are where you see both species of mussel. So these came originally, the zebra mussels, from Russia, and they were introduced accidentally when ships were releasing what's called ballast water. Ballast water they would release into the Great Lakes area, and they accidentally released some zebra mussels that were in that ballast water. The big problem with zebra mussels, they reproduce very quickly, but they particularly cause issues in that they can completely cover the surface of an area. They can even cover other organisms such as clams. Another big issue from zebra mussels is they can get inside pipes. They are small enough to do so, and when they do that, they can block those pipes. This is a problem for when you have wastewater treatment facilities, when you have nuclear power plants that are trying to draw in water, and so this becomes a very large issue. I'm going to show you a couple of images that will allow you to get an idea of just how pervasive and just how difficult zebra mussels can be. This first picture that's pulled up is an image of several zebra mussels in the hand of a person next to a dime so that you can see the relative size. Some of these mussels can get downright tiny. This next image shows a pipe that's been completely filled with zebra mussels that's been cut in half, just so you can see how packed these can be in a pipe and can impede water flow. Notice that's a person's hands around that pipe so you can get an idea of the scale of this. This next image shows you zebra mussels completely covering a screen that is supposed to be filtering water. And finally, just like I said that zebra mussels can completely cover up clams and other organisms, this last image shows a crayfish that has been completely covered by zebra mussels. Crayfish move, so the fact that these mussels can get onto it, attach, and start growing there is pretty impressive and scary. This is another slide that was not included in the printed notes. Once again, I recommend you pause, write it down. Fortunately for us, zebra mussels are pretty hardy, so anything that would be toxic enough to kill zebra mussels would kill off most of the life in a river, and it's very difficult to have just specific localized application of any sort of pesticide in a river or a body of water. It's going to spread and kill other things. So the best thing to do right now is to physically remove these from boats and pipes, or to put chemical coatings on anything that you'll have in the water that will prevent the zebra mussels from attaching. The problem with this is this can become very expensive, especially when you have buildings that have already been produced. And another way is you can actually physically scrape these off, or some hot water treatments can work as well. And last but not least, the scourge of the South, kudzu, our only plant invasive species that we're going to talk about today. Kudzu was originally brought in from Japan, as you can probably tell by the name, sounds remotely Japanese, and it was introduced in the late 1800s for a number of reasons. One, it was brought in as an ornamental plant to kind of make things look pretty. It was brought in for forage so that grazing animals would have something to eat, and it was also planted on a lot of hillsides to reduce erosion. Unfortunately, kudzu grows very quickly in the environments that you find in the southeastern United States. There's plenty of temperature, there's good temperature conditions, there's plenty of rainfall, and so kudzu started spreading out of control. This map shows the range over which kudzu has spread up until the year of 2011. So notice it's encompassed all of Hawaii, and it spread throughout the southeast all the way as far as East Texas. Here is yet another slide that I did not include in the notes, but I thought it would be a good idea to have some examples of how these invasive species are managed. A lot of the methods of dealing with kudzu have to do with cutting the plant or destroying the root system or the parts of it that grow so that the plant can't recover. So one method is to have livestock graze on the plant enough so that they'll prevent it from growing back. One website that I looked at recommended having 8 goats per acre to make sure that they graze the kudzu enough to kill it. You can also cut the vines just above the ground level, destroy all the cut material so that it can't then regrow if you, say, tried to compost it. You can also mow very closely, you can pre-burn areas, you can cut by hand, you can dig out the roots. There's also been some research into using Asian soybean rust as a biological control. It's a disease that afflicts kudzu, and there are several herbicides that I have listed here that you can also use to control kudzu. You don't have to memorize the names of these herbicides. I just put them on there in case you were curious. As I hope you've seen on the slides for all of the invasive species and how their populations are, or how we're attempting to manage their populations. There are a couple of different methods you can try and use to control these invasive species. You can use chemical methods, which involve using pesticides, herbicides, some sort of chemical that's toxic to that species, or you can use biological controls. If it's an animal, you can bring in a predator of it. If it is an animal or a plant, you can bring in a parasite or a disease-causing organism, or you can also bring in a competitor. There's just a danger that if you bring in another species that's not native, it too could have a population that goes out of control, and then you have another invasive species to worry about. In fact, that's what happened with the example that we watched in class, the cane toad, which was brought in to control the cane grub and ended up not only not solving the problem of the cane grub, but becoming a very large problem on its own. And now on to the topic that many of you guys informed us about, some of y'all in quite entertaining ways with your Endangered Species Project. Let's talk about those endangered animals and plants and other organisms. So the image that you see on the left here is showing something that shows up on the Wikipedia page of any animal species that you looked up. And it shows the conservation status that you have. Lists all the levels. You have extinct, extinct in the wild, critically endangered, endangered, and vulnerable, and then you have conservation dependent, near threatened, and least concern. Obviously, you guys know what it means when a species is extinct. When it is extinct in the wild, that means the only remaining organisms of that species are found in zoos. When you have organisms that are critically endangered, endangered, or vulnerable, without intervention, without something to try to prevent them from going extinct, they're going to go extinct in the near future. At lower risk, when things are conservation dependent or near threatened, you do have to make sure that you're taking precautions as a country, as wildlife management, to not degrade conditions for them further. And if a species is of least concern, you don't really have to worry about it. It's not going anywhere. For kicks and giggles, the image on the right is something that I found that's on a t-shirt that I think is hilarious. So it gives you some examples of each of these species. You have the extinct, which has dinosaurs, woolly mammoths, and dodos. You have the endangered, which has a species of cat, a panda, and a unicorn, which is apparently endangered. Threatened, which are lions and rhinos and apparently Bigfoot. Least concern, that's pigeons, humans, and cockroaches. And then what I find incredibly hilarious, the resurrected species, which are the phoenix, the zombie, and for those of you who are slightly blasphemous, Jesus as well. Not sure how clear the text on this is going to come out, but this is part of a list of endangered species. I believe it's from the IUCN, or International Union for Conservation of Nature, Red List of Endangered Species, and it has information about them. The status, whether they're critically endangered, vulnerable, etc. Also, what their trend is. The down arrows are obviously bad things are happening. Their populations are decreasing, and you have various notes and those who are assessing the populations of these species. You can find this information for any endangered species if you look hard enough. Begins a look at a couple of endangered species that I picked seemingly at random. You'll notice the bias because I have a couple of cats on here. Well, you guys know me. So, first, the Amur leopard, which you see a picture of. It's a very pretty kitty. It's located on the border, or in the area around the Korean peninsula, particularly where North Korea hits Russia. These animals are threatened by poaching, by habitat loss, and by inbreeding because of their small population. By the way, in case you didn't know, poaching is illegal hunting of animals. Now, the inbreeding is a big problem because there are less than 35 that are located, 35 organisms in the wild, but there are about 300 Amur leopards in captivity. Major problem is when you have animals in captivity, they don't always reproduce as well as they do in the wild. So there's no guarantee that just because you have a lot of animals in zoos that they'll be able to reproduce and you'll be able to create a viable population from them. Next, we have blue whales, which are threatened by hunting. They are the largest animals on the planet, and there are about 5,000 to 12,000 of them located in the wild. They're very large. There are obvious issues with trying to keep blue whales in captivity. If you look at the ocean map that you see on the bottom, the blue shaded area is the range of blue whales. So you'll see it encompasses most of the oceans on the planet. And their large size just makes it very difficult to think about keeping any of them in captivity. So any efforts to make sure that the species continues are going to have to be in the wild. Example I like to use often when I'm talking about genetic diversity and the risks of having very small populations and low genetic diversity is the cheetah. If you look at the map, the range of the cheetah is what's shaded in green. But in the past, before basically European colonization, cheetahs had a range that encompassed almost all of Africa and even parts of Asia. And now cheetahs are extinct in most of Asia. They're threatened by lots of things. They're threatened by direct hunting of them. They're threatened by habitat loss, fragmentation of their habitat, where their habitat's cut into smaller pieces, so different populations of cheetahs can't interbreed, and also reduced populations of their wild prey. There are about 10,000 of them in the wild and about 1,000 in captivity. And like I said, their low genetic diversity threatens their survival as a species. So in case you're not being tormented by commercials with very sad pictures of animals looking at you as if they're saying, "Help me, help me," here is your sad moment. Listen to the music and feel guilty. Okay, that's enough of the guilt trip. Now we're going to look at some of the things that are done to try and help endangered species recovered. Because it's not all sadness and despair. There are some species that have been brought back from the brink of extinction. Sorry, kiddos, this is yet another slide that you will need to write down from scratch. So like I said, stop the video, start a-writing. There are a couple of different major methods that are used to help conserve endangered species. One of the big ones is captive breeding programs. So animals are bred in captivity in zoos, in wildlife centers, and then they're released into the wild after a large enough population is built back up. One of the big limitations of this method is that some animals, when they're not raised in the wild, you have to teach them the behaviors they need in order to survive, in order to hunt, and in order to continue to live once you release them. Some of the notable successes that have been seen with this program are those of the California condor, black-footed ferrets, golden lion tamarins, which are, I believe, a monkey-type species, and red wolves. I know California condors were very successful in breeding programs. Another method that is used to try and help endangered species is private farming, where animals are endangered are actually owned by individuals. So the individuals will get the money from these animals and have, therefore an impetus to keep them alive because they can either sell them, sometimes, for example, as in the case of bison, they're sold as meat, or they can provide money through ecotourism. Scientists tend to worry about this, though, because when animals are raised in captivity and they're kept in captivity, they tend to become domesticated, and that involves behavioral and ultimately genetic changes in the species, which raises the question, are you actually saving the species from extinction, or are you just changing it? This has been done, as I said, with the bison and also the southern white rhino in South Africa. One of the more popular but difficult to maintain methods is to designate wildlife preserves. These are areas where these organisms' entire habitat has been preserved, and they're often owned and protected by the government. A major problem with this method is if you have poorer countries which have a lot of species that are endangered in them, they may not have the money to maintain these preserves, and they may not have the security to enforce to make sure that poachers are not coming on the property and killing animals. And then when you have endangered plants, you can have seed banks where seeds of various plant species are stored. In fact, something that we'll mention later, the Svalbard Seed Depository, which is in Norway, where they are trying to collect seeds of every plant species, both wild and domesticated on the planet. There are several important laws that relate to endangered species, both domestic and international, and we'll talk about those next. First legislation passed by the United States to try to help protect endangered species was the Lacey Act back in 1900. What it did is it prohibited trade in illegally hunted fish and wildlife, but the limitation was that it had to cross state boundaries for the Lacey Act to apply. This has been amended several times since to add new species, but there was a more comprehensive, better law that was put into place later, and that's our next topic. One of the three most important laws that we're going to learn about this year is the Endangered Species Act. This is a United States law that was passed in 1973. Now, the act is maintained and administered by the U.S. Fish and Wildlife Service and the National Oceanic and Atmospheric Administration, otherwise known as NOAA. Species have to meet certain criteria in order to be placed on the endangered species list, and once they're placed on the endangered species list, Fish and Wildlife and NOAA have to establish what's called a critical habitat, which is the most important habitat where most of that species lives, which is absolutely necessary to keep that species alive. They also have to create and implement a recovery plan to try and help the population come back up to sustainable levels. So several species have been helped dramatically by the Endangered Species Act. Most notably the bald eagle, our national symbol, the whooping crane, the peregrine falcon, the gray wolf, the gray whale, and the grizzly bear. Though there is some controversy now because lately the gray wolf has been taken off the endangered species list and hunting has been authorized, and there is not actually a large enough population to sustain hunting on the endangered species list. If you want to get involved, you can contact the Sierra Club, Conservation International, and a couple of other organizations. Here we encounter our first international treaty. So CITES, otherwise known as the Convention on International Trade in Endangered Species of Wild Fauna and Flora, was first ratified in 1975. It is a treaty that bans trade in endangered and threatened animals, and this is a very major protector of animals across the planet. So let's say that you are a poacher, you kill an elephant in Africa, and you're trying to sell the tusks to people who are making products out of it somewhere in Asia. If you get caught, then you can be basically punished under the conventions of CITES. You do need to know what CITES stands for. I have seen an AP test question that refers to it by the full name instead of the abbreviation. And only one species that has been listed on CITES has gone extinct since its implementation, so obviously it's being helpful. How do we know what numbers we have of a species to know if it's actually becoming endangered or not? Well, you have to gather population data, and we're going to talk about various methods for that. So oftentimes when you're sampling a population and gathering data, what you're doing is you're collecting live organisms. That's the most effective way to sample populations. But you have to sample them in such a way that you're getting representative information, meaning that you're getting information that represents the entire species. There are several different methods of sampling populations, and we're going to talk about each of them. By the way, if you're wondering what's happening in that picture, the scientist there is using a syringe to implant a tag in carp so that they can do a mark and recapture count. And we'll talk about what mark and recapture is on a subsequent slide. So with point sampling, you have a given area that you're trying to look at. You choose points on the map. They can be either random or you can space them out systematically, like the grid that you see on the bottom right-hand corner of the slide. Of the image. At each of those points, you're going to sample what organisms are located there. So you can use this to figure out species abundance, how many species you have, and the composition of the community in that area. Also, if you get a large enough sample size, you can start to develop some ideas about the characteristics of the population, what ages you have the organisms at, how they're reproducing, etc. We'll see about trying a couple of these sampling methods once the weather's warmer, so probably in the spring. Quadrat sampling is done. You pick an area in which you're going to sample, and then you are going to select certain smaller areas within that called quadrats. And then you're going to look at the different numbers of species you have. You're going to count the number of each of those species that you find in that quadrat, so you can get species density, you can figure out distribution of the species, you can figure out how frequently they occur, so how many of them you'll tend to have. If you're looking at plants, you can figure out what percentage of the ground is covered by those plants, and if you harvest plants from that area, you can even measure the biomass. If you're going to use quadrat sampling, though, you have to make sure each quadrat is the same size. Some people will make basically a large shape by taping meter sticks together, so they have a 1 meter by 1 meter quadrat. Other people I've heard of will actually just take hula hoops and throw them out in an area and use that as their quadrat. Now, you also have to make sure that your quadrats are appropriate to the size of the organisms that you're going to be looking at. So if you're doing a quadrat in a forest and you're trying to look at trees, tossing a hula hoop on the ground is probably not going to be your best bet for measuring the species abundance of trees. But if you're looking at species of grass, if you're looking at insects, you will use obviously a smaller quadrat. method of measuring populations, called a line transect, is really useful for trying to see how populations or how elements of the community will change as you physically move through the community. So you'll basically draw a line across a map, or sometimes they'll use tape or rope to mark out a line, and you'll record the species that occur along that line. You can either put these randomly so that you can just get a random sampling, or you can select some sort of environmental gradient, a slope, a change in salinity of the soil, whatever you're looking at, and you can set the line transect along that so you can see how the species is changing along that line. Belt transect is you're basically taking a set of quadrats, but you're taking them all in a row. So that's why it's called continuous quadrat sampling. You can get more information than on a line transect because you are actually looking at a wider area, but it's harder to carry out a belt transect. You're going to be looking at a lot more data. So you measure a strip across the study area, and then you use quadrats to sample at regular intervals along the belt. We're going to look at a couple of different types of belt transects on the next slide. So here are three different types of transects. You have a line transect shown at the top. You're just sampling at specific points along the line. The bottom two are examples of belt transect. So the first type is a continuous belt transect, where you are just measuring completely from one point to another in a wide strip. An interrupted belt transect is where you are measuring specific points along that belt. So you could, in this example that you see, be measuring species only in half of the area of the belt transect, which would make it a little easier. Trust me, I've done some quadrat sampling before, and if you are trying to count species in a very large area, especially if, say, you're looking at plant species and you're looking at grass, it would take forever to do a continuous belt transect of a lot of those species. So one thing to keep in mind is how much manpower you have in order to carry out one of these sampling. Another method that's useful, and it can be used to try to make estimates of population size and density, is mark and recapture. So just like the name states, you're going to mark some animals. Now, a lot of times this is used for invertebrates, small animals like the insects that are shown here. You capture a certain number, you're going to mark them in some way. The one way that I've seen done is you'll mark them with a little bit of nail polish. Not enough to kill them or hinder their movement, but just enough so you can pick them out. Then you're going to release them back into the population. You'll let them mix with the natural population. You may come back, say, a day later and do a second capture where you're going to now count out of the number of organisms that you've picked out, how many of those are marked. And if you get fewer back that are marked, you probably have a larger population of organisms. If most of the organisms on the second capture are marked, you probably have a pretty small population of organisms. This can be a little more difficult the smaller the animals get, just because you have to mark them in some way without killing them or injuring them. Fortunately, it's not always possible to directly catch or directly study organisms. Some of them, they are pretty skittish, they're hard to get a hold of. Some of them are spread out over very large areas, which makes it not very convenient and extremely difficult to be able to collect enough samples or collect enough organisms, and some of them are just darn hard to find. There's also the case that some of these organisms, you could cause them a lot of harm if you're actually capturing them. So you can use more indirect methods. So this is not going to be as accurate as when you can actually catch the organisms and start counting them, in person. So some of the ways that you can sample indirectly, you can collect scat, which means you basically are looking at poop. You can monitor calls that are made. This obviously is not going to work if you have more silent species. You can look at tracks like the paw prints that you see in that picture there, markings or scrapes, anything that's left behind that leaves evidence that that animal was there. You may be able to use electronic devices, motion detectors, cameras, etc., and you may be able to do measurements of how many burrows or nests or so on are there that give you an indication of how many of those animals might be living in an area. There are many ways to sample animal populations and try to gather animal populations so you can start counting up number of species. There are various nets that you can use in water. We'll look at some small mammal traps. There are some other methods that we're going to look at as well. Any of these methods that are listed here that we don't talk about later on in the video, you can actually look them up on your own. For example, we're not going to look at pooters, because the name kind of makes me laugh. No, that's not why we're looking at it. I've just never seen it mentioned on an AP test. Haven't really seen pitfall traps mentioned, and I've seen Tullgren funnels and Nansen bottles mentioned a lot more than kick sampling. So you're more than welcome to look those up on your own, or we may look at them in class. We're trying to study small critters, especially that are in soil or that are in leaf litter. You can use a Burley's or Tullgren funnel. I've only seen this mentioned on the AP test as a Burley's funnel, but you'll want to remember the Tullgren just in case they use the other name. So this is usually based on avoidance of something, light or heat. So what you have is a funnel and you have gauze underneath it. So that way little small creatures can actually climb through the gauze. As you see in the setup there, they have leaf litter containing the invertebrates, insects, worms, etc. that you want to look at. And then underneath that you have a collecting jar. So let's say you're looking at light avoidance. You shine a light on this leaf litter and all the animals that are trying to get away from that light are going to go down through the gauze and end up in your collecting jar where you can then count them. When the weather warms up, we'll see if we can collect some dirt or leaf litter and build ourselves a Burley's funnel. You're not going to find very much in the dirt right now. It's kind of frozen. There are several different types of nets that can be used to try and capture fish and other animals living in the water. So you see two pictures on the right-hand side. The top is a pike or a hoop net, where you have these hoops of decreasing size that are basically attached with netting. And so the fish enter and they're going to get trapped at the base, so that way you can catch them and look at them. Sign nets are actually pulled through the water, so that way you can trap fish in the netting and the mesh, and then you can count them. You also count invertebrates and other organisms living in the water as well, not just fish. Nansen bottles let you get water samples from certain depths and lakes, and they allow you to do it without disturbing the water too much. What you'll see is you have a tube that allows air to escape so that as the bottle fills with water, the air can leave. And then when you are done, you can remove the stopper. And when you're ready to collect, you remove the stopper and water's going to flow in. Notice there's a weight at the bottom to help it sink further in the water, and you'll lower it down to your desired depth. The picture that you see there is a man collecting purple sulfur bacteria from a lake, and you can actually see the purple, so you can see what they're collecting in that Nansen bottle. Now, as we move back to things on land again, if you're trying to trap small mammals, that's going to be harder than invertebrates. They're wily creatures, they're evasive, and they sometimes know that there's a trap. You can use Longworth traps, which are those little complicated contraption that you see in the picture there. Basically, it's going to have some sort of lure, and you're going to try to get the small critter to go in there. But some of these species are able to tell that you're looking at a trap. For example, this guy can tell it's a trap. When you are trying to figure out the range of an animal, like where it migrates, where it tends to live, a really good method to use is radio tracking. So you'll attach some sort of transmitter to the animal, and then you have a receiver that's going to pick up the signal and let you know the animal's position. There's a problem with this, though. As you can see from the images, you find this more difficult to do with smaller animals just because you don't want the transmitter to be bigger than the animal. Another thing is you want the transmitter to stick on the animal without harming the animal and without impeding its movement. Let's say that those penguins that you have pictured there were not able to swim as fast because of the transmitter. Well, you may not be able to look at their migration patterns because they may end up in the belly of a killer whale if they're not able to swim fast enough to escape. So you do need to be careful when you're choosing your animals that you're studying and your tracking devices. you can get some really neat data using radio tracking devices. So what you see here is a couple of tracks shown of great white sharks that were tagged in waters off the coast of South Africa. So what you see on the top one on the right-hand side, this shark traveled this path from South Africa over to Mozambique on the west coast of Africa, and they did that in 38 days. On the bottom, you see a much more amazing journey. You see a shark that swam from South Africa to Australia in 99 days. So then after that, within 9 months, they were back in South African waters. So they were taking basically a round trip of 20,000 kilometers. And you would not have been able to tell where these sharks were migrating without this radio tracking data, since it's not as if you can be in a plane and go, hey, look at that, there's that shark that we're following there. There's a couple of other methods that you can use to electronically try and measure populations. Electrofishing doesn't seem particularly nice to me. You basically are going to shoot an electric current into water, and as we saw with the giant Asian carp, the animals will be stunned and they're not able to move, but they're supposedly unhurt. I don't think that if you're trying to keep the population healthy and happy, that it's a good idea to go stun gunning them all the time, but oh well. Other electronic detection devices like the one you see pictured on the bottom, that is used to measure the calls of bats. So they will echolocate, they will make calls on certain frequencies. You tune that tracker in so that it will measure at those certain frequencies, and you can measure how many calls are being emitted basically per minute, per hour, and use that to try and estimate the numbers of organisms that you have in an area. This has been your handy dandy populations, biodiversity, human effects on populations notes. I hope you enjoyed them, and I hope there were not too many stray cat noises in the background. Thank you. Have a lovely day.